So this is our part 2 of latent representation of GenML models. Vedant is a research scientist, PhD in biophysics from University of Chicago.
So the value in a deep learning model is not in explaining things that we already could have figured out through PCA or something like that, but in explaining beyond. All of our protein sequences in the universe are drawn from some very particular kind of probability distribution. Imagine you have a coin, but you don't know the probability of that coin coming up heads. What is a way you could infer a coin will come up heads or tails? Adding more dimensions doesn't enable you any better of a function.
Okay, so it's 6:01 and we will get started as promised, and let other people join in, and in the meantime we'll continue with our discussion. So thank you everyone for joining. So this is our part 2 of latent representation of GenML models. And in simplistic term, in layman's term, it's like under the hood. So we want to like open up the box over here. So I made this picture using ChatGPT. So it opened up the box and kind of like dived into generative AI and see like what's going on. So our host is Vedant. So we had a fantastic discussion 2 weeks back, and the video is actually posted in Curious Circuits YouTube channel. And being said that this is my 18th episode of Generative AI in Enterprise Meetup Group. I started this group in December, and from there on, it has been a fantastic journey for me. So being said that, I'll just quickly move on to the agenda. By the way, there are new people who have joined in. So I'm Ujjal Bhattacharya, work in the financial industry, and I live in New Jersey. And this is just a fun thing for me, as well as I've made a lot of friends during this journey of this meetup group. So today's agenda is, of course, like we'll do the introduction, disclaimer, housekeeping, participant introduction. We'll jump into the topic of the day, and then we'll have some time for question and answers as well. So again, the viewpoints over here is, okay. So the viewpoints over here do not anyway represent the positions and strategies or opinions of my current employer. It's completely for fun, completely for entertainment purpose. We are meeting and we are having some fun together. And also, like, by doing that, we are gaining some knowledge. This video will be recorded and photographed and put it in social media. So if you have anything IP secret that you do not want to share, please don't, because we are going to put it in social media. If you have any concerns about appearing in the video, please turn off your microphone as well as your video. But again, like, feel free to speak up and talk, so that way, like, we can have an interactive session as well. Typically, I ask people to be on mute, and we'll try to wrap up by 40 minutes. So we'll do a quick round of audience introduction. I have mentioned myself, so I'll quickly go through the list over here. We'll leave our guest speaker at the end, but I'll go through my list over here. So I have Mar Aaron. Mar Aaron, we have met last week, but if you just want to give a one-liner intro for the other people.
I'm an electrical engineer, and I've been studying the architecture of the GPTs since early 2023, and I've written the apps, chat with PDFs. I'm interested in modifying GPTs, so I'm always trying to learn more about them.
Awesome. Okay, fantastic. So in my list, I have Vivek.
Right. My name is Vedant Sachdeva. I work in healthcare. I manage large initiatives, and we're looking to incorporate generative AI into a variety of our solutions.
Awesome. Jigar Joshi.
Hey, this is Jigar. Yeah, I work as a technical architect in one of the insurance company in New York.
Okay.
Yeah, I'm looking forward to explore more on AI front.
Okay.
Thank you.
Bahar.
Yes. Hi. Nice to meet you.
Nice to meet you, Bahar. Okay. Do you want to give a one-liner for like an introduction, or you want to pass?
Sure. I'm working mainly in natural language processing and large language models.
Okay.
I recently completed my master's and I'm looking for some job opportunities.
Awesome. Fantastic. Carlos?
My name is Carlos Belcastel. I'm an independent consultant, spending a lot of time, probably too much time, learning about large language models and AI agents.
Amazing. Okay. Actually, we have a meetup coming up on AI agent. So you have to watch out for that. It's fantastic. It's going to be on Crew AI. So just check that out.
Excellent. Thank you.
So did I miss anyone? Jion. Kim?
Hello.
Hi, Jion.
Yeah, hi. Oh, my name is Jeong Kim, and I am engineer, and I mostly worked on speech processing. Yeah, nice to meet you.
Nice to meet you. Okay, I think we have covered everyone. Let me know if I missed anyone. Good. Okay, so let's move on. So Vedant, those who do not know Vedant, Vedant is a research scientist, PhD in biophysics from University of Chicago, and has done a lot of work in collaboration with UCL and ENS Paris. So Vedant is our guest speaker today. He also did the presentation on this particular topic 2 weeks back, so that video is available in our YouTube channel. I'm going to shut up at this point of time and let Vedant take the floor and take us through the journey. So Vedant, if you want to do a quick recap as well, like what we talked about, so that we can have some context.
Sure. Hey folks, thanks for having me out here today. Before I share my screen, I'll go through a quick recap. So last time what we talked about primarily was the idea that generative models are largely looking to model the distributions and model distributions, and in particular be able to use to draw from those distributions to generate new examples that look like the inputs that we put in, right? So last time we talked a little bit about the idea that what we'd like to do is be able to model some variable, some response variable y, given an input variable x. And in particular, what we'd expect x and y to be are high-dimensional objects. We'd expect x to be a sentence and y to be the next sentence in that task, or x to be an image and y to be yet another image, or something like this. And in this conversation, one of the things we talked about is, okay, so this is sort of like a high-dimensional to high-dimensional mapping of some sort. What happens if you think about this as a high-dimensional to low-dimensional mapping, or more critically, a low-dimensional to high-dimensional mapping? And it is that idea of getting from high dimensions to low dimensions back to high dimensions, which gave us the ability to introduce autoencoders, and in particular, variational autoencoders. Variational autoencoders were introduced, I think nine years ago or so, by, I think it was Welling and Kingma. I think those are the authors on that paper. And it was maybe one of the very early implementations of a Gen AI model that people are quite familiar with today and maybe don't use so much anymore, and in like sort of cutting edge research settings, but are still quite interesting to look at. And what we'll be doing today is we'll actually kind of really look at one in some detail and try to understand what's going on with that. And I'll try to compare it to some of the well-known linear models that people are accustomed to from many years ago.
Awesome. So Vedant, I mean, like, so that was a good summary. I also was going through the recording one more time, and so if I look at it, in that particular discussion, you started off with linear regression at a very, very in layman's term, right? For me, so we started with linear regression and machine learning basics, like touched upon that. Then we talked about probability distribution in predictive modeling. Then you took us through a journey of Bayesian approach to regression. Then we touched about different kinds of generative models, and one of them is VAE. Right? And then we also kind of like touched upon deep learning and neural networks, and also some practical implementations that you are doing in your career right now in biophysics, where you are trying to generate some protein sequences out of the model that you have created, something like that. So did I capture it?
Yeah, so I think we'll go into some detail. We'll go into some detail on this. I think it's sort of one thing I will try to mention. We'll try to be somewhat explicit about it. It's like when I talk about variational autoencoders, I usually do mean variational autoencoders are a particular flavor of objective function of models these are usually implemented, the internal function approximations are usually implemented by deep neural nets. So you don't necessarily have to distinguish the two principles too much
I
when discussing them. Okay.
Okay. Yep.
Cool. Awesome. Feel free to ask questions as we go. I admit that today will be relatively technical, but I kind of wanted to be able to give everyone a flavor for how these kinds of things work. So we're gonna embrace the technicality and go into the weeds a little bit.
While you prepare this, this is what I was telling Vedant before you guys joined, that I did my mathematics honors 20 years back, 25 years back. I have no clue. I forgot everything. So this is a fantastic refresher for me as well. So Vedant, whenever you are bringing up this new terminologies, behind the scene, like after this session, what I'll do is like I'll take all these keywords and then go back and do Google, right? Search and try to connect the dots. It gives me a ability or a scope to refresh the memory and also learn a new things, a bunch of new things.
Absolutely.
So, amazing. Yeah, please go ahead.
So awesome. So with this in mind, we'll start talking about some of the observations that I've had in variational autoencoders over the couple of years that I've been using them. And as Ajal mentioned, I've largely spent I've spent a lot of time so far thinking about sequence-driven approaches for generative modeling in biology. And what do I mean by that? I mean, imagine you have a protein, but you don't have like a physical protein that is all twisted up and actually doing something in your body, but you have the sequence of amino acids that makes up that protein, right? Just the letters associated with those amino acids. One thing that you might find really interesting is, can you generate novel examples of such proteins just using the existing databases that are out there about those sequences? And there are many companies that try and model biology in this way, and they say that they will claim that like with these sort of approaches, you can generate novel examples of a protein that could be used for a therapeutic or sustainability purpose or something of this nature, right? You can solve some problem that exists out there at a molecular level. Now, these particular approaches, they leverage the idea that all of our protein sequences in the universe are drawn from some very particular kind of probability distribution. If I can learn the rules of those probability distributions, then when it comes time to generate a new example, I can just draw from that distribution. To maybe put it in a bit simpler terms, imagine you have a coin, but you don't know the probability of that coin coming up heads. What is a way you could infer whether or not that coin will come up heads or tails? Well, you could count the number of heads that emerge after 100 flips or something like that. That gives you the probability of the coin emerging heads. Now the question becomes, can you flip the coin again or simulate forward examples of possible coin flips that obey that kind of distribution, that parameter of that coin? Of course, this is easy if you have the coin. In biology terms, this would be like, this is easy if you could control all of evolution and could wait 1 million years for a protein sequence to evolve. But you'd like to do this on much faster timescales. You might do this without the coin. So giving examples of something that obey the rules of the distribution that we've learned is sort of what this model is after. One of the things that I'll be talking about pretty heavily today is I'll be comparing to early work using something called PCA, and its unabbreviated form, principal component analysis. If you're not familiar with principal component analysis, it is sort of an approach in which we take a look at if we imagine we have some big data set, let's say we have all of the 500 S&P stocks, and we have the time series stock prices of them as a function of time. And one of the things you'd like to do is say, you know what, like, I really think NVIDIA and Tesla, they tend to move together because they're all described by how much hype there is about AI at any given moment. And in fact, maybe there's a larger cohort of companies that are controlled by the overall hype there is in AI. And then there's another cohort of companies who is controlled by what the Fed's interest rate is. And another cohort of companies that is controlled by oil prices at any given time or something like this. The idea that you have these, massive high dimensional data set, these 500 different variables, but they're really only explained by a few. Principal component analysis is one of the early approaches for trying to tease out that underlying sort of explanatory power for that data. Now, one of the things that limits PCA, one of the things that limits principal component analysis is that it's constrained to only really focusing on the second order statistics of of the data set. What do I mean by that? Well, when we think about first and second order statistics, people usually talk about means, medians, modes, things like this. What is the central, where does the distribution kind of centralize around, or something like this? When we think about second order statistics, we start to think about variances and covariances, right? Like how wide is a distribution, and do two things move together or not? That's what people usually mean when they talk about covariances and second order statistics largely. So this is sort of a limitation. Thinking about second order statistics is sort of a limitation of sort of older work that in principle deep learning has enabled us to get away from it. And what we'll talk about is how that emerges and what it looks like in a VAE. Cool. So this I've talked about this in some detail with some detail already, but you know, the idea here is that we're going to use a VAE to model a bunch of protein sequences. And what we'd like to do is we'd like to, you know, be able to take a look at the spaces in which a sequence lands and somehow use some knowledge about that space to be able to engineer a better version of that sequence, because somehow that space, that small number of variables, represents something physical about the protein at hand. It explains something physical about the protein at hand. Cool. So if we take a look at a particular example of a variational autoencoder, here is one taken from a paper that was published in 2021. We can ask the question, okay, so if I fit this VAE to these protein sequences or something like this, and ask the question, where does a certain set of those sequences that bind to a very particular target land, you start to see that they cluster together, right? So these are the red sequences here. All the red sequences, these are experimentally measured, end up clustering in the same spot in latent space, while all the other sequences end up being elsewhere. So what this tells us is that if we want a new example of a sequence that binds to that kind of ligand, we should actually pick sequences from that spot, right? And because we know in general that, oh yeah, all the sequences that bind to this particular thing land in this area, when I draw from this spot and generate a novel sequence, I should get something that binds. And in practice, this hypothesis ends up holding up. This particular version is one paper demonstrating the ability to do this, but there's sort of been a lot of validation that this kind of approach is possible for generating novel examples that are functional in biology. If you'll remember from last time, I also shared this plot where we said, okay, well, if we have a variational autoencoder, let's take a look at another data set, another example data set. And what we saw was that you get this similar kind of separation of the characters of the data, the characteristics of the underlying data, right? So it's sort of hard to read, I guess, here because it's gray and black, but on the left-hand side, we see that all the protein sequences drawn from vertebrates end up in one part of latent space, and all the protein sequences drawn from invertebrates end up in a different part of latent space. We observe that all the protein sequences with a particular kind of specificity are all clustered together, right? The orange sequences tend to group together, the purple sequences tend to group together, the red sequences tend to group together, the blue sequences tend to group together, right? And this is occurring simultaneously, right? Like the two plots that are plotted here, they're actually both from the same exact model, plotted in the same space, just colored slightly differently for a representation of a different variable. You also can quickly, distinguish endotherms from exotherms, as I'm showing you in the right-hand plot. What is the value of this? Again, the value goes back to now when I want to generate an example of a very particular kind of vertebrate chymotrypsin from an endotherm or something like this, I can exactly specify to you where you should look and where you should generate that sequence from. So now the question becomes, how is this happening? What is the fundamental explanation for why this occurs? Like what is really happening when we look at a variational autoencoder and try it and really dig into the details there, right? So the first thing that you can ask is, well, we talked about PCA already. PCA is super well understood. You can ask the question, do VAEs learn second order statistics? Do they learn covariances? And one way you can do that is you can ask the question, okay, suppose we have some amino acid sequence, some candidate amino acid sequence. For those of you that maybe are very much into board games and card games and things like this, our amino acid sequence is very cleverly named Magic the Gathering. And what I'm proposing here is that, well, if we want to know how second order moments really emerge, what if we perturb the sequence at one point and see how the model responds at other points along the sequence, right? And if the model has really learned that there are second order correlations along this candidate amino acid sequence, when I perturb at one point, if there exists another site, another point that is highly correlated with it, that one should also want to move. So here, what I'm proposing is that, okay, we have some proposed sequence, magic the mathering. I've changed the G to an M or something like this. And, if we're thinking about English words or something like this, magic the mathering isn't a real word. So a model shouldn't, take in magic the mathering and give back magic the mathering, right? That's not a real example. But maybe magic the mothering or something like this is a real word. And so the G and A are correlated in some way. So when I move the G to an M, that means the A should also be thinking about moving. And so this sort of, correlated change in the distributions of the outputs are what I mean when I'm thinking about second order statistics in a particular model. Does that so far feel comfortable for people? This idea of like, oh yeah, things are correlated, and when I poke at one point, other things should change. Cool, awesome.
Question over here.
Yeah.
So you're giving the example of your domain. So
Mhm.
for example, can this be applied in any other domain as well? So for
Yeah.
example, in the financial
So-
industry or,
So I think, go ahead.
or any other industry as such. It will be more relatable if you can give another example.
Yeah, so I think in sort of like I think we can kind of go back to like more like a natural language processing example, and one of the reasons why I chose this particular sequence is because it's also in English, 3 English words put together, and so words themselves are correlated. If I say I climbed up a hill or something like this, that me saying climbed probably implies that the word mountain or hill or something like that is going to emerge. But if I said, if I instead had said, instead of I climbed up a hill, I said I broke up, you're not going to think hill belongs there, right? By changing climb to broke, you're going to think I probably broke up a fight or something like this. Or I broke up, I don't know, some wood or something, So in natural language processing terms, this is a kind of an example that you might think of. In financial models, you probably also have something very similar that like, okay, when some stock gets perturbed, let's say PayPal or something like this, probably other payment processing companies also feel some, exposure to that, because the people betting on payment processing companies probably they have some reason to believe one payment processor is different from another, but the sector as a whole is impacted by the same.
Yeah, we saw that in crypto like last month, right?
Yep.
Suddenly there was a surge.
Yeah, precisely.
Okay.
So these are the kinds of second order correlations that I'm thinking about.
Okay,
Cool.
makes sense.
And of course there are higher order correlations, but for now this is sort of a way of poking at the second order ones. So I have some full mathematical prescription here, but kind of the key takeaway here is like, if I really want to explore, at least in a very particular style, the covariances between things, I can just make 1 point perturbations and ask what else moves. What else moves when I make a single point perturbation? And that is sort of what I'm going to be doing here. And what you find is on the left-hand side, what I've plotted is a particular flavor of PCA called SCA. And this is effectively a covariance matrix.
Can you repeat the full form of PCA one more time?
Sure. Principal component analysis.
Okay, principal component. Okay.
Yeah, precisely. So this particular version is this particular style of matrix is called called statistical coupling analysis. It is sort of tailor-made for biological sequences by a set of scientists in 1999, Raman Ranganathan and forget his last name, but I cited him earlier. He was Lockless. Right. So this matrix is effectively a measure of covariances for data that is not continuous, right? And so that's what we see on the left-hand side. And on the right-hand side is the same kinds of covariances that you observe that the VAE learns. Now, these two matrices aren't one-to-one. They don't exactly match up. But if I were to do a decomposition on these two matrices and ask what are the most important domains in each of these matrices, we actually find that the two matrices start to agree that there really only are a few modes along which the whole data set varies. And so I think, good, I mentioned this exactly here, that the matrices don't exactly perfectly match up in all spots, but the most important features of both matrices are the same. And what that tells us is that at least for the most important variations observed by a variational autoencoder, they are what you get out of PCA. Now, this is a really good thing. This is a really, really good thing, right? Because if you got something that was dramatically different from PCA or something like this, you should be very, very concerned that you are missing things that you have seen to be important in the past, right? And you should be sort of skeptical that a model is missing those features unless you have some reason to believe that they were there for some reason that wasn't real or something like this. You were picking up on them without having a real reason to believe that they belonged. This kind of naturally lends itself to one early conclusion, is that some fraction of our deep learning models, our variational autoencoders, our transformers, and things like this are picking up on first and second order statistics. Those are things that we already knew how to solve back in the 90s, the 80s, the 70s, and the 60s, right? Like we already knew how to solve that problem. So the value in a deep learning model is not there, right? Is not in explaining things that we already could have figured out through PCA or something like that, but in explaining beyond what's beyond what is allowed, right? So
So let me ask
Go
a
ahead.
question over here. So basically, somebody over here is PCA with PCA or principal component analysis, and this one is a SCA, which is like more tending towards biology, right? Or
Precisely. Yeah.
okay. So basically, what you're saying is, and I might be wrong. Correct me if I'm wrong. But basically, what you're saying is like with PCA, this VA correlation matrix can be derived from PCA. Is that correct?
Well, it's more to say that PCA is one calculation to get at the statistics of the model, the statistics of the data that we are studying.
okay.
And this VAE correlation matrix, a VAE is another way to try and understand the statistics of the data that we're studying. And the core thing is that these two things agree to at least some extent, right? If they didn't agree to any extent, you shouldn't be super concerned that somehow something has gone wrong one way or the other.
I see. Okay. So basically, you're saying like whether I go with PC or whether I go with VA correlation matrix, both of them tend to agree at some point of time.
At least at some level, and this is kind of a nice result to have emerge, right?
But
Of the
then
ones
you're
that
saying
you
that
study.
with deep learning, I want to go beyond whatever we have already done because this has been like developed long back.
Yeah, so the PCA approaches have been developed a long time ago, so we should be able to get those with ease. It's about what comes beyond that, right? It's where they differ that becomes interesting.
Maredan, go ahead.
Concerning PCA, I understand that when they began turning words into vectors or an array of floating point
Yeah, you're thinking of like Word2Vec or something like this?
numbers. Yes, so they did that before GPT, and then they used PCA to map out that the relationship between words could be charted as directions. So if you went from king to queen, you would get the same direction as from man to woman or husband to wife. So
Yep.
PCA works with words that are represented as vectors as much as DNA sequences. Can I ask you a question? This DNA and VAE that you're talking about, do GPTs exhibit the same predictive properties with respect to the DNA sequencing, like A, G, T? If you fed in a sequence of DNA letters into a GPT, would it learn to give you another DNA strand that's valid?
Yeah, so your bog standard general purpose, like OpenAI, ChatGPT, foundational model or something like this, doesn't do this, but they're all
It's not pre-trained to do that. But if you
Yeah.
trained a GPT box with genetic information and you told it, this is a mouse, this is a dog, here's the DNA,
Yeah.
and then you trained it on all the DNA and all the labels,
Yeah.
would it be able to
There are
predict?
many such models that have been implemented. Genomic. So these fall under a class of models called genomic language models, GLMs, or genomic large language models. Sometimes people even refer to them as. And many such companies and academic groups are focusing on trying to utilize these to solve engineering problems. So
Have they proven any usefulness?
So I think there are a couple of papers that have demonstrated that you can achieve, performance improve, like improvements in the activity of a protein or something like this, or you could generate new examples of a particular flavor of protein. The Baker Lab is particularly famous for some of this work. The Ranganathan Lab is famous for some of this work. There's a host of people who like the Ross Lab. Mike, I think it's
So,
famous.
so do you think
Ross.
it will be possible for China in their DNA labs to take Brad Pitt DNA and Angelina Jolie DNA and some other person and then type in a command and print out a DNA that would produce another actor or another beautiful person?
So I think there are many challenges, many hurdles to try and make these kinds of things work. It would be super cool. I think just being at the stage where you could actually particularly say, I want this particular DNA sequence to yield something with this particular physical property is not a trivial problem at all. People have tried and there's been some progress made to this end. But one of the kind of core challenges is that it's really hard to do the experiments necessary to collect enough data to get enough labels to enable this kind of thing. And B, there are many, many, many interacting actors in any given biological system, right? Like you have the DNA sequence itself, but then there is the translation process where you go from DNA to RNA, and then there's RNA to, or I don't even remember if it's called translation. It might be called something else. It might be transcription. I don't remember. Anyway, but like you go from DNA to RNA and RNA to protein, and there are all these modifications that occur. And then even after the amino acid sequence is turned into a protein, there are post-translational modifications which change the protein sequence. And then it exists in the context of some more complicated biochemical circuit or something like this, where many things are interacting.
So you're saying it might produce something, but it wouldn't be viable necessarily? Is that what you're saying?
It's not guaranteed. I think there's a lot of variables that are not just generating new sequences. Yeah. So there's a good amount of complexity there that I think people are steadily working through. And the sequence, like the sequence generation and structure generation, like people are starting to make strides towards that, but there's many other variables that need to be solved out too.
So basically, you're saying, in another words, would say predicting a word is much easier than predicting the different genetic makeup.
Well, yeah. So one kind of reason in which it's easier is that you and I can stare at a word in whatever language we speak and be like, oh, that's a word, or that's a sentence, and that's a reasonable sentence, right? Like we could say, oh, I could imagine someone having said that or something like that. And as long as you have, right, like maybe I couldn't do that for Mandarin or for German or something like this, but like someone who does could tell me, right? Oh yeah, that's reasonable.
One of my friends tried with a Bengali translation or something like that. It was messed up.
Yeah, I'm sure, because many of these models are trained purely on English, right? But like the reason I bring up people who speak different languages is very much for the purpose that biology in and of itself, DNA sequences in and of themselves are a language, and the translator of that language are very, very complicated biochemical circuits, and it's not easy to understand what's like, make the translation, right? Make the translation from, oh, this is a sequence to, oh, this is a real, like biology could have produced this or something like this. That's not an easy translation for a human to make.
And the similar things are for video generation as well, right? That's what I was reading somewhere, that word generation, sentence generation is like a set of dimensions, but the moment it comes to pictures and then videos, now you have to imagine the whole sequence, which is like how you rotate a camera and then what other things. So it just makes it more complex. So of course, like DNA will be like even more tenfold, 100-fold, 1,000-fold more complex.
Yeah, absolutely. The complexities start to grow, and like even for video and image generation, like there are things that might look reasonable to you and me, but like to a more careful observer, like, oh, that hand has 6 fingers or something like this. And so suddenly something looks less realistic for this reason. So like the ability to evaluate the outputs of these models is not a trivial problem. It is a very, very challenging problem that many people think about quite a bit.
And that is one of the reasons like industries are failing to adapt.
Failing is that why do you say this?
Failing to adapt generative AI in a large scale. So
good, because it's not clear whether or not the outputs are reasonable. Yeah,
evaluation is the most hardest problem at this point of time.
Mm-hmm. Yeah, absolutely.
Okay,
cool. Awesome. If that's all good for people, what I'm basically trying to establish here is at the bare minimum, like we actually get back out PCA, which is super, super nice. it also kind of offers us the idea that maybe in some cases where we might think about using a deep learning model, but we haven't yet used a simpler, more straightforward model, using a simpler, more straightforward PCA type model where it is pretty tractable, pretty computable, pretty fast. It's worth doing, right? It's worth doing because you might get out a lot of the detail you're trying to explain through the use of sort of these linear, kind of straightforward principal component analysis style models. But that said, like, you do get these sort of like you can get a little bit more from a deep learning model than you can from a linear model. And what we're going to talk about next is a way in which we can actually determine how that works. And what I'm going to imagine is that, so to give an example of something that is not going to be explained by second order statistics super easily, we're going to think about this particular toy data set. Now, the toy data set right now has a lot of math. I'm going to try and break this down into sort of simple terms. And what I want us to imagine is, suppose we know the ideal sequence or the ideal set of sequences that biology should be evolving towards. Now, let's imagine that all of those sequences either point in this direction or point in this direction, right? They point in some existing direction. There's some vector of some kind. Now, if biology is allowed to evolve starting from a random point, it's eventually going to work its way over to one of these directions. And these general directions are what I'll call long motifs. But what's important, well, so this much detail you could probably get out from just any kind of linear model. Pretty much any model can solve that problem. What I'm then going to propose is that when you start to point near the direction of a long motif or something like that, there's actually a different rule that you need to obey. And that is going to be called the short motif. So maybe you get most of the way here, but once you actually are in this direction, there might be a reason to slightly tilt over to one side or something like this. So going back to maybe the example that Mar brought up about, in word to vec, going from man to woman, king to queen or something like this, man to woman and king to queen, that while the relative difference of those two words is quite similar, they actually exist in totally different descriptors of a system, right? Man to woman is super generic, but king to queen very clearly, you might apply when you're describing historical settings or something like this. So this is what I mean, right? Like there's coarse grained detail that gets out the gender of the gender of the target that we're describing, and there's fine grained detail. Now I'm going to be describing the short motif that captures a little bit more than you get from just that coarse grained detail. So imagining that we have a data set where we have control over both is what I'm going to be working with here. I'm going to say, imagine data organized this way. And I'm going to ask the question, can you get this out from a deep learning model? Cool. So, like I've said, this is kind of all inspired by some of the stuff I've seen in biological sequence data and things like that. So we'll hop sort of straight into it. And one of the things that you immediately observe when you build up a data set in this way is that you get a lot of agreement with what you had put in, with what you had observed in your biological sequence data, right? So here I plotted 4 kinds of important long motifs, 4 kinds of important coarse-grained directions in my data set, in my toy data set, which is on the right. And you see the data that lines up with one of those coarse-grained directions, they all cluster together. You get the blue, the orange, the red, and the green, I believe. These are all sequences that are quite close together, sequences that are quite similar, and they obey some coarse-grained direction that I, by hand, put into the generation process for that data. Why did I put it in? I put it in because that's kind of exactly how, you might imagine that biological sequence data has emerged. There's some coarse-grained idyllic sequences that most sequences point in, and if I pick that sequence and look around it, all sequences that are just like it also match up in the same kind of spot. So the blue is a cluster of sequences that is close together in the real biological data. The green is another cluster of sequences that is different from the blue that is clustered together. The orange is another cluster of sequences that is quite similar that is different from the blue and green, and so on, right? So kind of what I'm trying to say here is that this model at the very least captures the idea that this model at least gives us sequences that might have some, reasonableness to them, and that our model does the same kind of thing to these sequences, which is it clusters by similarity to other sequences. Now, one thing you can ask is, suppose I have many sequences that are very, very important, but I try to compress them down into a very small number of dimensions. Now, the more dimensions I have, you should imagine that it's super easy because you can allocate a dimension to each sort of ideal sequence that I put in. But if I have too few dimensions for the number of sequences for the number of patterns I might be putting in, the number of long motifs I might be putting in, the model actually has to figure out a way of organizing things without mixing things up. And so it sort of gets really like patchy and sort of like, oh yeah, these clusters sort of become really hard clusters where there's no kind of interpolation between them. You just have the sequences as they are. Here I'm referencing a paper from, I believe, someone who's now at Anthropic, was at OpenAI, El Hage. Hopefully I haven't mispronounced his name too horrendously, but he has spent some time looking at the interpretability of AI models, and what he calls this effect is polysemanticity, where one dimension in a model is used to explain more than one thing. And this is kind of what you're seeing here, right? You get one dimension explaining part of the green, part of the blue, and part of the black, and another dimension explaining part of the green, part of the red, part of the black, something like this. But critically, things that are highly uncorrelated, so one patch of red against another patch of red. So I should mention that like part of the way I've constructed this data is that there is one pattern, but for every pattern there exists an anti-pattern pair, which is exactly anti-correlated. And so the two separate patches of red correspond to a pattern anti-pattern pair. And the reason they're separated in the way that they are is because the model is trying to make sure it never confuses these two things. It never confuses these two things. So it tries to push them as far apart as it possibly can. Did you have a question there? Or?
And that is also because we have, and this patchiness, or like, the separation of this red patches in two different places. And that is the reason behind it's giving more dimension to this whole data set.
Well, right. So it's trying to find a way to keep things separate, even though it only has a few dimensions to work with.
So basically,
right?
Right. So I've given so many dimensions. So now they're trying to create a group or a segment, I think group, whatever you want to call it, in the patch, which has more correlated dimensions. The other patch of black, though it's black dots, but it still falls under a different set of dimensions.
Yeah, so when I'm talking about dimensions here, I want to be just we'll try and be super precise. I'm just thinking about the x-axis and the y-axis, right?
Oh, okay.
So yeah,
not like multi dimensions.
So right here, but here the data, there is at least 4 different kinds of pattern anti-pattern pairs that need to be explained by just the x and y axis. Right. So because it doesn't have an x, a y, a z, and a w axis to assign each of the pattern anti-pattern pairs to, it tries to make it work in just the 2 dimensions that it has. So it starts to like just push everything really far out to make it work. Yeah.
Had it been multi-dimension in this particular, all these different anti-patterns, maybe it will plot in a different way. But since you're plotting them into 2 dimensions, it kind of like tries to create that pattern.
Yeah, so actually this is you beat me to the punch a little bit. We will discuss in just a second, but this does actually pan out the way in which you're describing. Good. So one of the things that I might then ask is sort of like, okay, so I have this sort of modeling approach of like okay, I have this fake data. I know exactly how likely any given example is from this fake data. What happens if I start generating from this model? So this is the question, right? Like the model generates reasonable sequences, but are all reasonable sequences generated, right? Like imagine that I have some sequences that, the model never sees for some reason. Let's suppose that reason might be that like I've never exposed my model to science fiction or something like this. I've never exposed my model to this particular style of speaking or something like this. I've never exposed it to that kind of thing. If a model is really good, it should still be able to generate things that belong to that group, right? Like if the model is really successful, then it can generalize outside of the domain it's just specifically trained on. If it's really learned the probability distribution well. And for this toy data set, things are so simple that I think it does quite a reasonable job at generating sequences, generating pretty much all available reasonable sequences. There are some things that don't quite get represented as well as you would have liked, but for the most part, things get generated quite well. That said, good. I forgot to mention this detail. That said, the difference between the left and the right here is that the left is the limit where there are more patterns than there are dimensions. And in that limit is where you start to generate, is where not all reasonable sequences get generated. You generate things that look way more like the data than the data really should, then should really reasonably be allowed.
Mark has a question. Yeah.
Yeah, go ahead.
I had the hand up from before, but there's enough vectors in GPTs to separate a lot of things out, but do GPTs pattern things this way also in clusters?
Yeah, so I actually this I'm not actually super sure of. I haven't sat down and like tried with a transformer to like look for this kind of organization. Actually, that said, there are papers that suggest pretty heavily that transformers tend to actually pursue this kind of prescription of data. So the paper in particular that I'm thinking of is Hopfield models are all you need. Hopfield being J.J. Hopfield, who developed an associative memory network model back in like the 1980s or something like this, which has still had a lot of power to today. There was a group in like 2023 or 2022 or something like this who demonstrate that transformers actually do basically learn these kinds of patterns in data and just try to allocate everything to one of those patterns. But it's a slightly different, like it's slightly different than the mathematical formulation of an associative memory network that I proposed here is different from what they believe the associative memory network that is learned by a transformer is. So there's a slight difference there. Slight is maybe an understatement. There's a big difference there. But yeah, so I think this is a great question because it does beg the question, like, how reasonable is any of what I'm saying? How general is any of what I'm saying? And I think the answer is that there's at least a good amount of evidence that suggests that what I'm saying has some amount of generalizability, that you could get similar kinds of observations across models, across models that are used today. And they also kind of challenge some of the notions about generalizability and things like this, because, you know, you have to ask the question, like, are the patterns you're learning really useful patterns for generating new examples? And this is sort of what this slide is kind of getting at, is that if you have too few dimensions for too many patterns, you probably don't generate things that are, you probably don't generate the things that you don't see enough of in your data set. You generate things that look too much like your data, and you can't generalize to new examples. So this is to say that a model that is only ever trained on nonfiction books or something like this would struggle to generate a fiction book or something like that. It would struggle to create the notion of magic or something like this. Of course, you know, hallucinations are a thing, and models are not very good at knowing whether or not they're real. But the idea is sort of what I'm trying to get at, is that like it can't learn a new style, or it can't describe a new style, even though anyone should be able to say like, oh yeah, that new style could totally be possible. Cool. So with that said, I did also kind of give you, I did also work through the case of like what happens when you give too many dimensions for just a few patterns. And what ends up happening in this case, for this very particular data set, is that you get two dimensions that basically get allocated to describing nothing. They're just noise or something like this. And you get another two dimensions that are just describing the variation that is important. So you kind of see this in this plot here, where I plotted all the different slices, all the different pairs of 4 dimensions, and 2 of those dimensions, latent dimensions 2 and 4, give us back that kind of like very ordered, oh yeah, right? This is where the patterns are, and here's the axis along which they lie, and here's the combinations of the patterns, and everything else just looks like a noisy mess. So that's kind of a nice observation to have in your back pocket too, that like models can also be kind of know what the true dimensionality of the data they're trained on is. Cool. Part of what this also asks though is like, okay, well, like Vedant is saying all of these extra dimensions are noise or something like this. Shouldn't we be able to compress those dimensions out? Is it dangerous to have those dimensions? Can we have a model automatically recognize not to encode those dimensions? And in the particular prescription of a variational autoencoder that I presented here, you actually can't do that. The models the dimensions start to become redundant, and this kind of leads to instability in the generative process. Sorry, I'm going to skip this slide a little bit. But if you choose a different flavor of variational autoencoder called a beta VAE, where basically you change the importance of matching some Gaussian distribution, some prior distribution, you actually get a model that, like, in an automated fashion, recognizes the number of dimensions that belongs to the data, and like quickly and sharply picks up new additional dimensions as you put it in. So that's what this slide in some detail, in like some very limited detail, is showing for us here, is that you can kind of quickly have a way of sort of automatically detecting the dimensionality of the data you're training on. Finally, the last thing I'll talk about is this idea of Simpson's paradox. So for those of us who are not familiar with Simpson's paradox, this is sort of a fun example. And let's see if I can set it up properly. Where, how is it? Okay, sorry. I don't remember the toy example off the top of my head, but there are all sorts of cases where something that sometimes happens is that, like, you get two academic departments which are trying to achieve, parity in, let's say parity in their age groups or something like this, and they're both trying to get a little bit younger and hire some younger people or something like this. And one department is like X% old and the other department is Y% old, and both increase in percentage individually by how young they get. But when you pool the groups, you end up decreasing overall. And this is because of because by pooling the groups, you actually get this sort of like weird lack of stratification that occurs, that creates this illusion that you've actually gotten worse, even though at an individual level you've gotten better. It would be useful if I could actually remember the toy example that everyone talked about. I can't remember right now. But there's a Wikipedia page on it is quite useful, and I would strongly recommend you take a look. I might just do that right now. Good. I think this plot is sort of useful. I pulled it up on my phone. Let me just maybe I can just stop sharing my screen and pull it up very quickly on the good. So hopefully you can see this kind of funny plot from Wikipedia, where if I kind of blind myself to the colors and I tried to explain the variation on the data, I would say, oh, look, the data is downward trending in X, right? That's what the average of the two groups is. That's because these two groups themselves are actually quite different from one another, and I shouldn't try to describe them by the same model. And so you get this reversal of a trend by segmenting out your data. There are very famous examples of this for like trying to achieve gender parity in academic departments and things like this. So this is called Simpson's paradox. And the reason I bring up Simpson's paradox is because when I talked about my short motif, long motif, short motif, long motif, the short motif, long motif stuff that I was describing earlier, one thing that you might imagine is, oh, look, the long motif might get described really, really well, but does the short motif get described well as well? And in an ideal world, with your model, you would manage to describe both perfectly. In practice, at least in VAEs, at some point you sometimes don't see that emerge. You see cases where you don't get the short motif at the cost of the long motif, or you do get the short motif at the cost of the long motif. And so it becomes useful to have sort of tiered hierarchical models that separate these things out. So this, I think, actually could be kind of thought of as a sort of fine tuning or something like this. I haven't thought about it that way thus far, but it is kind of maybe something that is kind of a possible example of fine tuning. But with that, oh, good. Why was this idea important? Why was this idea useful? Because it seems to work for proteins too. It seems to also emerge for proteins, that by segmenting down your data and hierarchically selecting and fine-tuning on a very specific part of your data set, you actually do better in your regression task. You do better in describing the details of that. So you might have some long motif that explains a lot of the variation, but those short motifs you don't get until you fine-tune. So with that said, I'm actually going to stop here, and I'll stop sharing my screen, and I will take any questions that people have. But that would hopefully what you saw from all that is kind of a hopefully somewhat interesting set of observations, set of ideas that exist about variational autoencoders that show you here are some things that a variational autoencoder captures. here are some of the things it doesn't capture. Here's maybe a use case for fine tuning, and here's perhaps maybe that use case falls apart, and things of this nature. Really is what I was kind of going after and exploring.
I have a quick question for you, right? So is this, so thinking about it, right, from folks who are trying to select different models for, a business application.
Mhm.
Does this help in evaluating, right, depending upon how many dimensions are being used from the variational autoencoder perspective, right? How to evaluate which model would be better suited for their purpose? Does that help in that direction?
Yeah, so I think if you imagine that like one of the problems you're super interested in solving is like you're training some suite of models on some data set and you're trying to select the best model or something like this, this kind of approach is sort of valuable because what you get out is sort of like a more parsimonious model for explaining your data without having to risk overfitting or risk underfitting or something like this.
Right.
So it should be clear when we're talking about this, what we're imagining is a variational autoencoder with 1 dimensions, 2 dimensions, 3 dimensions, 4 dimensions, 5 dimensions, and so on. We're not necessarily talking about comparisons of variational autoencoders to transformers to diffusion models or something like this, though it is conceivable that something like this could be achieved.
Interesting. This was like jam-packed with so much of good information. I think I'll need a day to process. Hey, Martin, go ahead.
I had ChatGPT help me write a neural net to implement the XOR, the Boolean XOR 2 input problem, and it was supposed to and it had a pre-training system, and it did work in the sense that it had a reducing loss, and it generally output a number close to 0 or close to 1 when it was supposed to. And I tried to increase the number of neurons in the neural net, and it didn't really make it any more effective or efficient. I don't know. Is that a theory that if you add too many neurons, it just stops working? Is that what you were showing?
So I think so what you're describing here is like, okay, in some limit, an XOR gate is sort of simple, right? Like you have 2 inputs and you get 1 output, and all you need is some way of functionally approximating the rule that like the sum of these 2 things has to be, if the sum of these 2 things is 1, I get 1, and if the sum of these 2 things is 0 or 2, I get 0, right? That's what the neural network is trying to approximate in that case. The reason why adding more neurons in that particular limit might not do anything is because you already have come quite close to approximating that function that gives you 0 if you put in 0 or 2, and put in 1 if you put in 1, right? What I'm showing is sort of maybe is somewhat related in that saying like adding more dimensions doesn't enable you any better of a function. So that is I think part of what I'm showing is exactly that, right? Like that you have some function you're trying to model, and giving your model more parameters doesn't necessarily enable it to model that simple function any better. That said, like for things that are super, super complex, like natural language processing, like video processing, I don't think people have yet come to enough parameters where they've run into this problem. And in fact, OpenAI and has spent a lot of time arguing that like actually more parameters is always better. So there's something interesting there to explore at a theoretical level that I have not yet appreciated in full detail yet. The theory I think is not yet solved out.
Do you know of a simple Python a folder that has a set of Python scripts that implement different types of neural nets that you can just run and see what happens and then modify them and things like that? Is there like a free collection of those things? I mean, that example, the XOR, it's something that they do in college, but I had the GPT write it from scratch. But is there a whole suite of these things that you can just experiment with?
So it depends upon like so I think both TensorFlow and PyTorch have like really nice tutorials showing like way like the actual like foundation websites that correspond to TensorFlow and PyTorch, right? They have really nice tutorials showing ways of using different deep neural nets to do something. Like MLP solves this task, LSTM solves this task, recurrent neural nets solve this task, convolutional neural nets solve this task. And they also have sufficient inbuilt library, like the libraries are sufficiently built up that you can just kind of like with a few lines of code change from working on multi-layer perceptron to working with a convolutional neural net to working with an LSTM or something like this. So I think looking in that direction is probably useful. I was thinking that it might be fun to do a workshop seminar where we like code up a model together or something
Yeah.
like this.
Let's do that.
But like my general advice for someone trying to like learn these things for the first time is like find a model that you like and know super well, start use that, and then just like where you use torch.nn.linear or something like this, replace that with torch.nn.convolve1d. Maybe I don't remember if that's the right syntax there, but or torch.nn.lstm or something like this, and just try those kinds of things. That's like how you might plug and play different styles of deep neural nets.
So if someone wants to learn more in detail, right, about this particular topic, is there any reference material or any thing that you can recommend?
Yeah, so
A course or something like that?
yeah, so it depends, right? Like if you're looking to just learn how to code some of these models up and things like this, the advice that I would give is to like pull up a GitHub of a model that someone else has already done and just like read and type out line by line yourself exactly how this thing works and try and like suss out what every line is doing. If you're more interested in sort of the mathematics of these models or something like this, I think that like
Yeah, from the mathematics standpoint, right?
yeah.
If someone wants to start from scratch, for example, myself, right?
Yeah, so let's see if I can pull
where
this up. This one is really popular. And I think it's reasonably accessible, but they do tend to like hop towards more. They tend to have a bend towards physicists.
Okay.
Oh, sorry, hang on. Let me just drop it in the Slack. Drop it in the chat. Sorry. This one. This tends to be pretty reasonable, and I think is actually accompanied by a GitHub that like shows you every single example of code that they're working through. Oh, yeah, cool.
Okay.
This is quite nice.
Definitely, I'll give it a shot.
Yeah,
At least
and if
try.
there's other like Andrew Ng obviously has good coursework. If you like, wanted to
deep
follow
learning.
Andre Karpathy or something like this when he posts his various tutorials on making nano GPT and things like this, those are also
Okay.
useful.
Awesome. So 7:05, I think we'll wrap up. Vedant, thanks for your enthusiasm and excitement while teaching us.
Yeah, hopefully this was somewhat informative and that everyone got something out of it.
I loved it. Okay, until then, so 2 weeks from now, we are going to catch up and we're going to continue from where Vedant has left off. Thank you so much, everyone. Take care. Good night.